Amino acids are the building blocks of life, intricately involved in protein synthesis and numerous other biological processes. This presentation comprehensively explores amino acids, delving into their structure, classification, and functions. Amino acids are the basic building blocks of proteins. All amino acids have in their structure a central carbon atom, called the alpha carbon. Also, they have an amino group, a carboxyl group, a hydrogen atom, and a side chain, all bound to the alpha carbon. The difference in the side chain group or R group determines the unique properties of each amino acid. Amino acids can be classified based on their side chain properties. They can be polar, nonpolar, charged, or aromatic, nonpolar hydrophobic. The nonpolar hydrophobic amino acids lack side chain polarity. They tend not to interact with water molecules. These amino acids are glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Polar hydrophilic. These amino acids are water-loving because of electronegativity differences between atoms in the side chains, like oxygen or nitrogen. Their presence causes a polypeptide chain to twist toward water, while a hydrophobic chain causes a twist away from water. The polar hydrophilic amino acids are serine, threonine, asparagine, tyrosine, and glutamine. Acidic amino acids. You get an acidic side chain if a carboxylic acid is in a side chain. At basic pH values, carboxylic groups can be charged negatively. Amino acids with acidic side groups are aspartic acid or aspartate and glutamic acid or glutamate. Basic or positively charged amino acids. Basic amino acids have a nitrogen atom with a lone electron pair that can react with a hydrogen atom. At a low pH acidic conditions, the amino acid may attach to a free proton to form a conjugate acid salt. Identify a basic amino acid by the presence of a positive charged nitrogen in its side chain. The basic amino acids are lysine, arginine, and histidine. During the 19th century, chemists, mostly in Germany, purified and analyzed the then new family of amino acids. By the beginning of the 20th century, 18 amino acids were identified as components of proteins. As each amino acid was described, the discoverer gave it a name depending on its source, properties, or chemical structure. Except for the suffix "in", there was no systematic naming of the amino acids in the name of most amino acids. As the study of the amino acid evolved, writing out the full names of the amino acids became tedious. A joint nomenclature commission of the International Union of Pure and Applied Chemistry and the International Union of Biochemistry examined the names of the amino acids, along with the abbreviation of the amino acid names to the first three letters of their names. Soon, the three-letter abbreviations became tiresome to write, and a group of Czech biochemists proposed a one-letter abbreviation system. A 20-letter amino acid alphabet was designed and adopted by the IUPAC-IUB Joint Commission on Biochemical Nomenclature. In nutrition, amino acids are classified as either essential or non-essential. These classifications were established based on studies highlighting that certain amino acids were necessary for growth and nitrogen balance, even when others were available. Essential amino acids are those that the human body lacks the metabolic pathways required to synthesize, so these amino acids must be supplied from an exogenous diet. In conclusion, amino acids form the fundamental building blocks of life, contributing to the intricate machinery of biological processes. Understanding their structure, classification, and functions is essential for comprehending the complexities of protein synthesis and the broader realms of biochemistry and molecular biology.
This video is part of the course Metabolic Fundamentals of Obesity at the University of Oaxaca, Mexico.